Simple. We'll see if I'm successful or not. So the lab basically is about transformation, which is moving DNA from empty space, basically from the environment, into something, usually bacteria. And it was a big question whether DNA would work in different places. So we're setting up this experiment to try to prove that. And what makes it difficult is that you need a lot of controls to prove that what you think is happening is really happening. So the first one in the top left is we're just going to grow bacteria normally, right? So we add bacteria to a petri dish. We add food source, which is what that LB is, a nutrient broth, and bacteria will grow. Piece of cake, right? No DNA has been added. None of these plasmids that carry DNA. It's just regular bacteria. And the second one in the upper right, still no DNA has been added, but we're going to add something called ampicillin, which is an antibiotic, and that should kill the bacteria. And guess what? It does. So you try to grow bacteria in that, you give the bacteria food, but then you give it an antibiotic, it's dead. Right? The other two we're going to add the DNA to. We're going to add the plasmids. And really the third one that does not glow almost proves our point. Right? So because this plasmid we're adding gives it ampicillin resistance. So we add the DNA, we try to grow the bacteria in a nutrient broth, the LB, but also in ampicillin, which should kill it, but the DNA we gave it resistance, so it grows. And you see those little spots on there. But really, we don't care about the ampicillin resistance. We really care about this glowing. Right? So what we do is now we add the chemical that turns on the glowing gene. It's a sugar called arabinose. And that's designated as the ARA. So if you put this bacteria in with the plasmid that carries resistance to ampicillin and glowing, and you give it the ampicillin won't die, and you give it the sugar arabinose, it'll cause it to glow, and there it is glowing. Right? It's a lot, and the controls are all there to set up like, wait, what is really causing it to glow? If you just grew some bacteria, it doesn't tell you much. If you grew some bacteria that grow in the ampicillin, it doesn't tell you much. You really need to prove that it's that they're taking on DNA that makes them glow, and so that's what they're trying to do there. This lab, has been simplified over the years to not use the arabinose, so you might see versions out there without it. It's still kind of proving the same point that if you use DNA with a glowing protein in it, you can get bacteria to take up that DNA and then, in fact, glow. You actually can give this to the embryo of almost any organism and cause it to glow, including a monkey or whatever. Generally, we do it so that you know a different gene is happening. So you attach it to a gene that's important in, say, fetal development of the heart. And when the heart is developing, it will also glow, and you've kind of successfully used it as a marker. Anyway, I hope that helps, and please don't hesitate to contact me again if you need anything.